Okay, I think we're going to get started. Good morning, everybody. Happy Mother's Day. I'm Jennifer Keller Jackson. I'm a Senior Program Officer at the National Collegiate Inventors and Innovators Alliance. Wow, I feel like I'm on, this huge stage in this huge room. So as people come in, bring them closer to the front so it doesn't feel so weird. Because from behind the bright lights, it's a little funny to be in such a big room. If you're in this room because you're interested in ideas going from initial idea, invention, innovation to impact, and you believe that this can happen, raise your hand. Okay, excellent. How many of you are actually working on an idea and trying to take it out to the real world? Okay, that doesn't mean poster session or paper. It means actually out into the real world. Okay, great. That's great. So NCAA, my organization, is a national nonprofit based in Massachusetts, and we support everybody working at the intersection. Okay, I lied, not everybody. Some people working at the intersection of technology, innovation, and entrepreneurship. So we work with a lot of students and faculty and also global innovators who are grantees from USAID and the Gates Foundation. And we provide funding and training for students and faculty and these global innovators to take their ideas out to market. The rock stars that I've assembled today who are on the panel, I've had the honor of working with for the last couple of years, supporting their work. And so there's really kind of two stories here. There's the academic program, the education and the training that these ideas are nurtured in, and then there is the actual project and usually the student-led innovation that is in some stage of getting to market. I guess I should have asked how to advance the slides before I got up here. I'm going to try this. Oop. Slides. How do I advance the slides?
Is it clickers?
Oh, you know what? I'm left-handed, so I was clicking the wrong side. So just to set the context before we will hear from Yusuf Yazdi, Sean Monagle from Johns Hopkins, Mohammed Zaman, Doresh Desai, and Grace Wu from BU. I just want to set a little bit of context about our conversation. And those of you that are coming in, make yourselves comfortable and come to the front so that we can see you. So NCAA works with, as I said, about 200 universities across the US, many of whom have partners and relationships with international universities and NGOs and partners outside of the US. Am I not allowed to advance the slides? There we go. We funded about 400 different courses and programs, and about 3/4 of them are still ongoing. So we find that our very small, very early seed money goes into these courses and programs that then become part of the institution and keep going. Out of those programs, 180 ventures have been started in the last 5 or 6 years. And when I say venture, I'm really speaking very broadly about nonprofits, for-profits, licensing deals, and other partnerships that get these innovations to market. So here are some of the logos. About 1/3 of our portfolio is global health, healthcare, medical devices, and then within that is global health. And then we have clean tech, new materials, and IT and agriculture, so I'm really talking about technology writ large. So I was on a panel on Friday where we talked about the challenges, and really instead of putting up the 18 or 20 challenges, that I brainstormed, everything is challenging. I mean, absolutely everything about this work is hard, and the challenges are not what we're going to talk about today. We're going to talk about how to overcome the challenges, whether they're technical, whether it's team, whether it's funding, business model, sustainability, et cetera. I'm sorry, I'm here. Whoops. Go back. Can you go back? I like this concept that Krista Donaldson from Brilliance, which is a product developed by DREVs that many of you may know out of Stanford. We're talking about the valley of hard work instead of the valley of death. And I think that's where these superstars are all working, in the valley of hard work, where we are going from the idea, invention, innovation, to what all of us funders up here talk about, impact scale. This is an example. I'm going to tell you 3 quick stories, then I'm going to get out of the way and you're going to hear from the panelists. 3 quick success stories. I'd love to do, the 30 failure stories that are the story behind the story for all of these. This is Krista's product called Brilliance, which is a phototherapy device. It looks like a reading lamp that can be adjusted up and down. There's no incubator or, cradle attached to it. And it's selling in India at about $450 right now. They launched last November. They were back-ordered at the factory within 2 months. Advancing, hello. Thank you. Krista is fabulous because she sends me, this is every funder's dream, every couple months, she sends me an email or I can go look on her website. This is just a screenshot of her website, her impact numbers. So they've actually treated 16,000 babies. They think they've saved the lives or prevented brain damage in over 300, and they've got 427 units installed since last November. And they're growing, and they have other products besides this. Second story. We made a small grant of about $18,000 to Rice University to support an infant CPAP machine. You all know what that is, right? It's an infant respirator. And Jocelyn on the left was a student at Rice who got that grant, and this is a picture of her standing next to Rebecca Richards-Cordum, her professor, when they got a $2 million award from USAID at the Gates Foundation in Seattle about a year ago. So that was amazing to see that small investment turn into something that then funded clinical trials, which have just been completed. And now they have their device, which you'll see on the next slide, is in 8 hospitals in Malawi and growing to 27. And there's the Pumani machine. The final story I'll tell you is an innovator named Ashifi Gogo, who was a PhD student at Dartmouth, he's in the foreground here, who launched a company called Sprach. It's going to sound a little bit like PharmaCheck that you're going to hear about later. And they have, they're tackling counterfeit drugs, and they've started in West Africa, in Ghana and Nigeria, and also are working in India. And this is not a manufactured product. This is basically a software play where you buy some pills, there's a label on it, like a scratch label, like a lottery ticket, and you scratch it off, you get a unique number, and you text that number into a phone, and you're told whether it's authentic or not. As you all know, this is a huge problem. Depending on what study you read, it's about 40% people in West Africa going to pharmacies getting the wrong drugs. And the next slide shows you this is a screenshot from their website a couple days ago. The top right hand is about 9 million verifications so far. I don't know, I tried to find out what percentage of those were inauthentic drugs versus authentic drugs. I don't know that number yet, but they have, it's starting to spread. So I'm up here to tell you that this is possible. We see it happen. And whether you're talking about scale at 100,000 or 100 million, we are starting to see really exciting results from what takes way too much time and money and work from a lot of these students and faculty. So I'm going to stop. The next slide just has my contact information. If you want to know more about NCAA or our grants programs, our training, feel free to email me. I won't respond on Mother's Day, but I will respond this week. And I'd now like to turn this over to Yusuf Yazdi. Dr. Yazdi is at Johns Hopkins, and he's going to introduce the program that he runs, and then we're going to hear from his just recently former student. I have to stop calling them students, especially now since most of them have PhDs. And we're going to hear about both the educational program and then an example of the technology, and Sean brought some show and tell items too. Yusuf?
I'm supposed to figure it out. Could I have the slide deck? Let's see. Let's start from the beginning, perhaps. That's Sean Monagle's slide deck. This one says Yazdi, I believe, at the beginning.
But it's the right school.
Yeah, close. We're getting there. So thank you very much, Jennifer. We've really been honored to be a partner with MCAA. Many of the projects I'll be talking about today are just, I won't actually have time to talk about them, but I'll just show a brief summary of all of our global health projects. We're funded in part early on in the process by NCIA. Nope, not this one either. The title of the file is Yazdi, CUGH, I believe.
It's not there.
That looks promising. Try that one.
They may be on the same, if they go down, it may be on the same one.
Yeah, maybe farther down. Well, let's not waste time. Well, it's really an honor to be here on Mother's Day. A lot of our projects, I would say maybe 8 out of, maybe 80% of them are related to mothers and newborns, and congratulations to all the mothers in the audience. Congratulations to all of you who spend your professional lives really focusing on helping mothers and newborns. I don't think there's anything greater in terms of humanity. If humanity has value, then probably the most valuable part is that moment when mother meets baby. And any effort we can do to make that a happy moment is worth spending our professional lives doing. Our center is a center within the Department of Biomedical Engineering. It's called if you had a slide, it would say the Center for Bioengineering, Innovation, and Design. I'm the executive director. The center includes an undergraduate program and a master's program, and now a couple of PhD students, and many fellows and folks who've stayed on and worked on projects beyond that, and then moved on to our partner organization, JPIGO, for global health projects, or moved on to startups. In fact, this year, about half of the class is staying on in Baltimore, working in some capacity. We're also very closely partnered with other entities that are affiliated with Johns Hopkins, like JPIGO and the Center for Global Health and the School of Public Health. I'm honored to have our colleague Yuka Manabe is also in the audience. I apologize if I don't recognize anybody else because of the bright lights, but we also have projects in TB, which emerged from our program. The mission of CBID, this is still not the right slide. If you want to keep trying, that's great. While I talk, you can keep going. The mission of CBID is twofold. It's about education and innovation. So Jennifer asked that we focus on how do you translate all this great ideas into actual use. And I have this phenomenal slide. It's just mind-blowing. You can't see it right now, but it's just amazing. And the basic premise of the slide is this. So let me go back to the Center for Global Health, our center, what our mission is. It's twofold. It's about education and it's about innovation. So I know that slide that Jennifer showed in the early ideation phase and proof of concept phase, we spend a lot of time at that phase. And I think that's where the root of the success downstream has to be planted. A project that's successful downstream is a project that's had a wide range of perspectives involved early on in the process. If you wait till later in the process to find out, how will this device get cleared by the FDA? How will this device get uptaken by Ministry of Health someplace? It's too late. Every project should have some degree of understanding through interaction with experts on all of the downstream issues. Hey, awesome. Thank you. So I'll push the next slide. Wow. So the metric of our success is impact. But it's impact through both the things that we do. Global health through the education of the students and also through the innovations that they develop. And we feel like the two are not really at cross purposes. And we've had many arguments, and I came from industry, so having a mission statement was natural to me, but it wasn't necessarily natural to all the faculty. But the idea of having a mission statement that was more than education was also a little bit of controversial, believe it or not. But we had a firm belief at CBID that students learn best when they're working on real-world projects with real implications, where they have a real passion. Because if you go into our design studios, you'll see students there day and night, literally. There's a couch there, they spend the night there, there's a coffee machine there, and Sean and his classmates, when they were students, you would find them there all the time. It's because they really are working on something they have a passion for and they want to solve. And the reason why they have that passion is because they've been to the field and they've seen patients dying from diseases needlessly, and they really want to have an impact. So each year we do about 20 medtech innovation projects. About 8 of those this year are global health related, dealing with over 100 students. About 40 clinicians work with us on our projects. And I think if you added up the potential benefit of this whole pipeline of projects, it's in the millions of people around the world. We do projects both for, we do projects that are medtech related and global health related. So we're not a global health center. We're really a center about medical device innovation. Let me say a word about that. This whole conference is about global health, but I believe that the border or the barrier, the psychological perhaps barrier between global health and medtech innovation, or medtech or healthcare for the developed world, is rapidly fading and becoming more and more academic. In the year after I arrived, we started our global health innovation program. Before that, it was all about medtech innovation for high resource environments. And we debated a lot of whether we should make this a separate track, so half the students would do global health, half would do medtech, and it was really our firm conviction that students, engineers, if they want to be leaders in this field, they need to know how to innovate for both of these worlds. In fact, these two worlds are really not two worlds. And there are many examples of innovations developed, for example, a low-cost blood glucose meter developed for India at 1/10 the cost of the device that's used in the United States. And this happened at J&J. And now that low-cost blood glucose meter is right there at CVS alongside the more expensive one. And it's taken up 70% of the market share for blood glucose meters. And the beauty of that is that we are not here to serve necessarily the needs of people in other countries. We're here to partner with people in low resource environments because we have as much to learn from them, and we have as much to benefit from in working in those extreme low resource environments for our own healthcare system here in the United States, as they have to benefit from our presence and our partnership with them in bringing best practices from the developed world, from medtech innovation companies like J&J or Medtronic, to global health. So I think that learning goes both ways. In fact, I think eventually that barrier between the different healthcare systems and the types of innovations they need is fading rapidly. We had a low-cost contraction monitor project this year. An undergraduate team was working on it, and they started running the numbers and doing interviews, and they realized that this was developed for sub-Saharan Africa to coordinate with a device we have called the ePartogram. But they talked to ambulance drivers in the US and they said, well, we have no easy way to monitor the contractions of a mother we're transporting to the hospital for delivery, and that could be a fantastic product for that. There's not room in the ambulance for a toco dynamometer. You need something much smaller, easier to use, maybe not as accurate, but that's a lot better than me putting my hand on the abdomen of the mother. So that's a fantastic example, I think, of an innovation that was designed for the global health, but is going to be probably very useful here as well. So this is the fantastic slide. Now it's a disappointment, actually. You actually see it. But this is the slide that I was referring to, and it's really all about bringing in, just to summarize it because time is short, to successfully translate a project all the way through to impact in the field. All of these stakeholders are critical. And you can't wait until late in the process to involve them. So we, in our process, this is just a philosophical slide, really. Our product development process, of course, is much more refined than this. But the idea is basically this. Let's say you start at that green dot with a clinical problem, and then you traverse the little blue line like Dorothy and her friends, and you get to that yellow dot there at where it intersects the first vertical axis. And you do a reality check. You stop. you make sure you've covered all the bases on the clinical, commercial, technical design, and organizational strategic issues before you progress. It doesn't mean you do a full analysis for a couple of $100,000 of the regulatory pathway of a certain technology early on in the process. That area under the curve there, under regulatory, for example, is just maybe a couple of hours of conversations. But you need to have that conversation early on. For example, we had a project that could have been a Class 3 device. This is in our medtech world. And conversations early on with regulatory experts helped us to redesign it with no loss of functionality, redesign the way the messaging of the device is communicated to the surgeon to make it a Class 2 device. That's a huge hurdle that's now been reduced, and a huge accelerator of the project to the field. How am I doing on time? 4 minutes, okay. The same thing goes for all these other stakeholders. You don't have to have many, many conversations and do a clinical trial early on in the process. You have to have a few conversations. You don't necessarily have to build huge, complex technical prototypes. You just have to have enough to get to that first stage gate, that first yellow dot, and incorporate all the perspectives you have in your design. Then if the project still makes sense, you go a little deeper on all those. You continue for another loop around the spiral, going deeper and deeper on all of these different areas of topics that are critical. By the time you get farther along in this path, you'll have an innovation that not only looked good at the beginning, but through constant iteration and refinement and interaction with critical stakeholders, it actually has a higher likelihood of success. And you won't have a lot of these downstream failures or the dusty shelf of great ideas that's typical. So here's a quick snapshot of projects of the last 3 years that CBID has done in the global health. Of course, this is just the subset that has to do with global health. I've highlighted the ones that are actually still active right now, and I put in parentheses some of the funding sources for the individual projects. We've been very fortunate to have tremendous support from Laerdal Global Health, which is a nonprofit subsidiary of Laerdal Medical, and they have funded several of our projects. They've funded our students to travel overseas. We also had funding this year from Medtronic, a very unique kind of partnership where the CEO of Medtronic said, I want your teams to help us figure out why people at the bottom of the pyramid in India who need pacemakers are not getting pacemakers. And I don't want you to tell me that we need a better pacemaker. I want you to go there and figure out exactly what's wrong. So our students went to India. 4 or 5 of our students, 4 of their engineers, Medtronic engineers, together spent a month in India. They went back a couple of times after that, and they really interviewed hundreds of hours of interviews with people in the field, rural medical practitioners, others, and found out exactly what is the root cause in this whole care pathway of people who have bradycardia, sick sinus syndrome, other conditions, are not getting the treatment they need. And they focused and developed a fantastic innovation, I think, that's actually in full development now with an NGO partner in the West Bengal. And others as well some are in full development, and some, stop. In academia, it's kind of hard for people to stomach the idea that my line of research or my project is done. But that is absolutely essential. If you have a portfolio of projects and you want success, you have to be willing to cull that list on a regular basis and focus your attention on those that actually have a potential to go forward. So if you'll see, there's a lot of here that says, several projects that say development halted, and we're actually proud of those because we do want to be able to focus properly our attention. So I won't go into the individual projects. I'll leave that up to my now colleague, Sean Monagle, and I'll stop right there.
All right, can I get my slides? Thank you. So again, my name is Sean Monagle. I was a student in the CBID Master's Program, graduated in 2011, so almost 3 years now. And since then, I've actually been working on the project that that I worked on in the CBID program. I was hired by my company, Japaigo, to continue that development. But before I get into that, I wanted to discuss quickly another project that we are also developing called the Cryo Pop. And this is addressing cervical cancer, specifically in the developing world. Cervical cancer causes about 500,000, or there's 500,000 new cases every year, and about 250,000 deaths as a result of cervical cancer. And this is actually projected to increase almost exponentially over the next 20 to 50 years. And JPIGO has already started to implement a program called the single visit approach to, the single visit approach to address this. And basically what they are doing is they actually take women, and you can screen for cervical cancer using acetic acid, which is basically just vinegar. So you can rub vinegar on the cervix, and precancerous lesions will appear as basically little white dots. And if you see those, you can treat them right then. And what JPIGO has started to implement is cryotherapy treatment. So they'll screen them right there and then treat them with cryotherapy, basically freeze the entire surface of the cervix, which kills all of the precancerous lesions before they can develop into cancer. And JPIGO has already started to implement this, but there's an issue with the cryotherapy equipment that they actually use. Primarily just expense and efficiency. So the current technology is about $1,500 to $2,000, and it's not very efficient. An entire 50-pound CO2 tank can only treat about 15 to 20 patients. So what Martone and his team have developed is basically a new cryotherapy applicator, so it'll still use the same tanks. And the way the current cryotherapy device works, you can see at the top there's a little metal tip, and it takes carbon dioxide gas from the gas portion of the tank, and it just constantly cools that tip to make it really, really cold so you can freeze the cervix. What they're actually doing is basically developing just a basic plastic. It's a plastic applicator. Most of the pieces can be injection molded for much cheaper. And they're actually taking from the liquid portion of the carbon dioxide, so you're actually getting a piece of dry ice. The entire applicator will be filled with just dry ice, and then you can press that up against the cervix for treatment. And just by making out of plastic injection molded parts, that can bring the cost down. They're estimated about $200 per unit. And that's also expected to drastically increase the efficiency. Rather than constantly using gas to cool the tip, you just fill up the tip with actual dry ice and use that for treatment. And one of the exciting points about this project is the business model is already in development. As I mentioned, JPIGO, the company I'm working for, has already started to implement this program just with a more expensive, inefficient cryotherapy technology. So if they can get this technology developed and to market, there is already somewhat of an established business model to get this out there. Next slide. Oh, I think I have that. And here are just some of the specs of the CryoPop. Martone was going to be here, but he's actually in Kenya right now doing a usability assessment on their most recent prototype. So no actual clinical treatment. They're just using the current prototype with nurses in Kenya on a model to see how well they can use the device and if there's anything they can improve and anything like that. Next slide. So this is the project that I actually started, as I mentioned, I actually started this as an undergrad at Johns Hopkins, and later worked more on in the CBID master's program. And what we were developing was a low-cost screening test for preeclampsia and eclampsia. This is the second leading cause of maternal death in developing countries, actually across the whole world. And one problem we identified early is treatment, while a problem in developing countries, isn't the biggest problem with this condition. Treatment actually is available in many areas, not all, but in many areas of developing countries. But one thing we found is that the actual screening for preeclampsia is inaccurate and very expensive. So currently what they use right now for basic screening are the urine dipsticks. So those are the basic strips that you pee in a cup and they'll dip the stick in, and the different pads will change color. Well, there's some issues with the accuracy of that, the color change, even though it comes with a color chart. There's a lot of inaccuracy with that test, as well as the price. The better dipsticks cost about 20 to 40 cents per strip, and that's too expensive for wide-scale use in developing nations. There are some cheaper dipsticks for about 5 to 10 cents each, but those are even worse than the good ones. The accuracy is not good at all. So what we developed, the first generation of our prototype was essentially a ballpoint pen you can see in the top left. And what you would do is you would take this, a health worker could mark on a piece of paper and give that to a pregnant woman, and then she can urinate directly on that, like a pregnancy test, and if the color changes, that indicates positive for protein, which is the symptom we're looking for. And if they're identified as positive, they can be referred to another health clinic or a hospital for further care. And we've done a lot of work on the actual reagent that's inside, so it's similar to what's in a dipstick, but we've done a lot of work to make it a more dichotomous test, so rather than a graded change that you get in a dipstick, it's more of a yes, no, just a positive, negative. And also just by manufacturing it this way, rather than manufacturing individual strips and then shipping them, basically putting the reagent in a pen and providing the paper, the health worker basically becomes their own manufacturer. So this, we expect to drastically reduce the cost down to about 1/2 to 1 penny per test. And as I mentioned, so this was our prototype that we worked on in my undergrad and graduate years. And the sponsor of this was JPIGO, the nonprofit organization that CBIT and JPIGO, or Johns Hopkins works with. And I was actually, after I graduated from CBIT 3 years ago, JPIGO actually hired me to continue development on this. And we actually got to travel out to the field. We had already been to the field once during the CBIT program, as Dr. Yazdi mentioned. And I got to travel out again back in 2011 to actually run field studies on this pen prototype that we had. So we got 600 to 800 women recruited. We actually had health workers make test strips and give it to the women. And we assess the usability of the technology, like making the strips, women peeing on the strips, as well as the accuracy, sensitivity, specificity. And it went well. We identified a couple key issues that we needed to address. For example, the health workers were fairly competent at making the test strips, but there was some variability. The strip needs to be made in a relatively consistent manner. And so in order to help them make the strips a little bit more consistently, we transferred from the pen prototype to what you see at the bottom left is essentially just an eyedropper. So now rather than marking different strips, you just put one drop and you're good. But you can see through the pictures here, I mean, working with JPIGO has been great. We've been able to get actually out to the field and test it pretty quick. We've now been out there 3 times. I actually just came back from spending 2 months in January, February, running studies on this new dropper concept. And again, everything went well. We identified 1 or 2 issues that we need to address, but the issues are becoming easier and easier to deal with. And moving forward, we hope to have our final prototype by the end of this year, and then at that point start working with a manufacturer to start production sometime next year in 2015. I think that's all I have. Oh. I do have some prototypes of both the CryoPop and our droppers, so feel free to come up afterwards, and I'd be happy to discuss more about them.
Thank you to both Yusuf and Sean. I think what I admire so much about their work is the complexity of the simplicity that they are doing. So I remember getting the proposal for the CryoPop and thinking, okay, this sounds really complicated. How are they going to get all this CO2 in the middle of somewhere, Guess who else works with CO2? in East Africa, for example? Any guesses? Coca-Cola, bingo. Pepsi. So, you know, I just remember that from the very beginning. They were thinking about, you know, using those local resources and trying to make things as simple as possible. And then going from what we thought was a very brilliant, simple design of a pen down to, you know, a plastic kind of eye drop bottle, which is probably even simpler and cheaper to make. So that's amazing. So we have time now. We're going to take a little break from talking at you and ask anybody who has a question to go up to one of the microphones that are around the room, and we're going to ask you to direct your questions to either Dr. Yazdi or to Sean about either the educational program at Hopkins or the some of the projects, or specifically the CryoPop or the antenatal products. And if you guys don't have questions, I'm going to ask a couple myself. So I'll give you a second. And
I have
go.
a question.
Where are you? Oh, there you are. Okay.
So,
Thank you.
do you have any issues with the university where the student is enrolled wanting a piece of the pie after it goes into production and is successful?
Is that a question for me or for Hopkins?
Anybody?
I can answer kind of globally, since we work with 200 universities. I can tell you that of the 200 universities we work with, there are 200 different IP policies in general, and most of them are very hard to understand. But NCAA does not have a problem with that. In fact, we encourage student-led projects, and we encourage where it's possible. It's not always possible with graduate students, but it usually is possible with undergrads to keep a piece of that pie. But it is something that is between the University Tech Transfer Office usually and the Inventors. Do you want to add anything? You guys have mics that I think are on.
Yeah, just very briefly, the trustees of Johns Hopkins have decided that if you do a project in the course of a classroom, that you own your own IP, unless you're an employee. I think it's the same at BU then. And but the faculty and employees like Sean are not, of course, their IP is owned by the university. But students, about 90% of the cases at Hopkins transfer their ownership of their IP to the university or assign that because the university provides excellent service in preparing the patents, et cetera. But we have had several cases where students have gone on their own and developed that, and we welcome that and we support them either way.
So Sean, are you building your retirement fund out of projected?
I don't think on a low-cost one-penny test, but we do, and my entire team actually is on the patent for the antenatal technology, and you know, if there are any returns on that, it'll be divided amongst all of us.
Okay, Mohammed?
No,
Oh,
that's fine.
yeah.
Just a couple of questions for Sean, and I also concur about the way that you took something complex and made it so simple. It seems like, wow, how come it was never thought of? But I guess I have a question about both of the two technologies you discussed. With the CryoPop, are there issues with morbidity because of the potential of burning healthy tissue around the involved areas? I would think that might be a major problem. And with the protein test, do you get a lot of false positives just from things like orthostatic proteinuria? Have you developed a way to really get the higher concentration of protein to be the kind of deciding determinant in the color change? Thanks.
Sure. So first, for the CryoPop, that definitely is an issue. The good thing is right now the cryotherapy technology is out there and being used. So what Martone and his team are trying to do is just to mimic that same technology. So to keep the same level of ice and freezing to occur as it does with the current technology. And I know that's a big driver of a lot of their designs. Just having the applicator tip, they spent a lot of time working on the diameter of that. Just because if the diameter is too big, exactly what you just said would happen. A lot of the surrounding tissue would get frozen as well. So that is a big design driver, I guess I would say. They've done a lot of work looking into that. And then for the false positives, yes, we have done basically a lot of our work is trying to find that good threshold where to have the positive and the negative colors. And a lot of our field visits are trying to fine-tune that threshold. Well, first to fine-tune that threshold, and second, to make sure the health workers, what we see is like yellow. So our test goes from yellow, and then it's either green or blue for positive. And a lot of our work is A, making sure that threshold is correct, and B, that what we see is yellow and green, so do health workers in developing countries. That's something that we didn't even anticipate when we first went out there, is a lot of different countries have, honestly, different definitions of color. I think it was one good story, like when I was out in Nepal, Nepal really only has eight or nine colors, which may be confusing, but basically what I mean is here, if you see a green, it could be light green, it could be dark green, it could be teal, it could be aqua. There's probably like 10 or 20, and heaven forbid you go to Home Depot, there's probably like 100 different greens. But that doesn't really exist out there. Like something that's green is green, something that's blue is blue, and actually they have a lot of trouble identifying colors in the middle. One of the health workers showed me that she got a nurse and then was pointing to different colors in the room. And I'm a Jaguars fan, unfortunately, but as you know, the Jaguars have like a teal color, and I was wearing a teal shirt at the time, and they pointed to my shirt and said, "What color is that?" And she couldn't say, like she didn't know what color it was, because it wasn't green, it wasn't blue. So it's just fun, interesting things like that you never learn unless you actually go out there.
Just a very quick question. Wonderful technology is amazing. Who trains the people in the field to use this accurately? So how do you get training of those who are going to implement the technologies in real lives of people?
Yeah, so we're really fortunate to be working with JPIGO, and now I'm a JPIGO employee, and I should have spent a little bit more time on them during the start of my talk, but they're actually a nonprofit. They're present currently in about 50 different countries and have worked in about 150 different countries. They've been around for about 40 years and currently about a $200 million a year organization. So they're very big, very expansive. And a lot of what they do is maternal and child health in developing countries. So they've already implemented a lot of training programs, a lot of distributing essential medicines to remote areas in developing countries. They've had a very successful misoprostol program for postpartum hemorrhage. So we're actually really excited that once we get these products to to market, once they're through regulatory, JPIGO has years of experience in training health workers and training nurses and using this equipment, which we're very fortunate to have. We just need to get those products through to the market.
And JPIGO works with or partners sometimes with non-Johns Hopkins people, correct?
Yeah, so it is a Hopkins organization, but they work a lot with USAID, WHO, World Bank, all of those organizations.
But would they work with the University of Michigan, for example?
Oh, yeah, sure.
Okay, just checking for you. Okay, great. Other questions?
Hi, my name is Shing. I'm from Albert Einstein College of Medicine, and I actually did my master's at Hopkins. Great inventions. I have a question. This morning, keynote speaker mentioned Global Health 2.0, where we're trying to empower more of our global partners. I was wondering if you could give examples of things that were invented locally and are actually being transferred back to the West, and or any advances in training the local workers in identifying and sort of coming up with new ideas instead of us sending people there and patenting inventions here and then transferring technology there.
I could mention one example. The one I mentioned during my brief presentation was a low-cost blood glucose meter that was developed by Johnson & Johnson's India division. So, and then that ended up being used in the United States. So it took a device that it cost like $50, $60 average sales price with maybe 20 different buttons on it to a device that the sales price is about $10 to $15 and has only one button on it. So there are many examples like that of products that were developed for, I just can't think of any other ones right now, but I would say multinational corporations, we're not all necessarily fans of multinational corporations, but having worked for one for 13 years before I came to Hopkins, I realized that despite all the negativity associated with that type of institution, it also has some positive impact in that they are present in many, many countries, and they train local people, and they develop products for those local markets, and they do have a natural mechanism to distribute those and for that innovation from India or China or Vietnam to end up coming back to developed markets.
I'd just like to add one
Sean,
more
do you have
example.
any other examples?
Oh, sorry.
Just one real quick. I know Japaigo is trying to develop something that they call the community cooker that was actually developed by a group, I forget what country, I think Kenya or Ethiopia. But basically it's a process, it's like a local village. Some villagers came together and they were taking all of the trash that was just everywhere and started burning it for fuel, and they started to use that to provide hot water for showers and cleaning dishes and everything, clothes, and also using the fire for as fuel or for cooking to make dishes. And Japaigo has heard about that, and they're starting to work with that group to try and develop that to actually train other villages and make that into a program where they can train people to do stuff like this, keep it clean, only burn what, should be burned, not releasing any toxic. But I do know that's one innovation that we actually, Japaigo, just kind of stumbled across and they found, and they figured that's a good idea, so we should try and develop that more.
Add to that, just very briefly, another way to accomplish similar objectives is to bring students from developing countries to come to a program like Hopkins. So this year, for example, we have 9 of our students are foreign students, and we had through Laerdal Global Health, we had scholarships, and we were able to attract 3 students, female students from Africa, one from Botswana, one Cameroon, and one Kenya, to come to the program. We have 2 students from Thailand, and those were, it's relatively new to us that we're getting more and more students from underdeveloped or developing countries. And I think that's a natural way. When they go back, they'll understand the process, they'll make the connections here, and they could become champions of doing similar programs back home.
So in addition to what Yusuf just mentioned, there's also a Stanford India biodesign program where they in fact bring fellows from India for 6 months to Stanford to teach them their biodesign process, and then the fellows go back home. And so what they've done is built this huge network of innovative docs who are now steeped in this process and really energized to go out and find, solutions to problems. And they've also built a curriculum around it that's going into several of the IITs in India, so that it's not just you have to fly, to Mountain View to get this training. So I think there are lots of examples of that. We have time for one last question.
Yeah, thank you very much for this very interesting panel. I'm going to make a comment and a question, but from the point of view of an implementer, okay?
Please.
So, an implementer. My name is Patty Garcia. I work in Lima, Peru, Cayetano Heredia University. And actually, when the first speaker after you presented this beautiful figure that goes from the idea into all the cycles and then commercial, one of the things that and I would like your opinion, what I haven't seen there really is implementation, which goes after commercial really, which is a big deal because there are several products, and we work with one of them, like the rapid syphilis test, for example, that were out there, but people were not using it, okay? And basically, and it was not necessarily that it was not simple enough to be used, but it's not, I think it's, I would like to see how can you add maybe, or comment, how can you add that in your picture? Because it's not only the participation that has to be part of the design, so the user participation, but also there is a whole process of implementation that goes into myths, beliefs, training, and other issues that sometimes we don't know, and that can lead us to think about innovation as a multidisciplinary approach also. The developers, the implementers that had to be there. With the comment about the reading, one of the things that we realized, for example, not only about the color issue as a cultural issue, but also as a biological issue, we learned that something that people don't think about, like color blindness, it's relatively common. And I'm glad that you're talking about green or blue. I don't know how much, but for example, for those tests that give you a red, we found in an evaluation in all the laboratories in Peru that we had 1% of the lab technicians were having color blindness. 1%. And they had been working there for a long time, and several of the tests were black or red. So this implementation phase, how can we approach and how can we work more closely with developers? And how can we make really global, not only to have developers there and implementers there, but to have this interconnection so we can have implementers in the country also.
Who'd like to answer that? Go ahead.
Thank you so much. That's a wonderful comment, a very great insight. And that chart that you saw was really from the developed world, advanced world, medtech development perspective. That's how we started, and we've been gradually modifying it to make it more and more relevant in both the philosophy and the terminology to global health innovation. And I think we missed the point that you raised was really important. I really appreciate it, because I think the side that says, the sector in the lower right that says commercialization is really also about implementation. Because in the US, commercialization involves, what does it take, all the stakeholders that are required to take your idea all the way through and develop a sustainable business around it. That's the capitalist or the US or the advanced world model. An analogy to that would be, or the analogous mechanism would be something pertaining to implementation through NGOs and through other organizations. So we do get some of those perspectives from when we interview clinicians, we interview health workers, community health workers. That comes up in the clinical sector as well, but I think you're absolutely right. We need to have perhaps a separate chart that really focuses on the terminology and the processes of implementation for global health. So thank you very much.
And maybe a whole separate panel just on that topic. So thank you. We're going to go up north now to Boston University, and you're going to hear from first Mohammed Zaman.
Thank you so much, Jennifer, and thank you so much to all of you for coming. So first of all, I won't take up too much time because I think the meat of any of these conversations is always in the Q&A. I'll have my students talk a little bit. So we're going to have a little bit of a change of pace. So we'll go from the undergraduate students who do remarkable work at many institutions. Hopkins has led by example for many years now, and BU is trying to follow in their footsteps, to graduate students who are looking at these things as thesis questions, who are looking at these things as a combination of research and implementation. So that's the first thing we'll do. The second thing I want to sort of say is NCIA has been there for us well before there was anybody else, perhaps well before there were friends and family. So we are really deeply grateful to NCIA for taking a chance on these projects, and you'll hear more about one of them in detail. So we are really, really grateful for that. Now, just as a show of hands, how many of you are not card-carrying engineers? And so neither am I, for that matter. But that's actually exactly why we are here. So I was trained as a chemist, and I'm in the Department of Biomedical Engineering. I don't think my dean knows that I'm not an engineer. But anyway, but the point is that these kinds of questions require exactly this kind of audience, that there are people who are coming to us from anthropology and public health and medicine and sociology and economics. Because at the end of the day, the implementation phase is certainly dependent upon sort of all of us working together. So what I'm going to talk about today in just 3 slides is a little bit of a personal story and how I got involved in something here. I've been thinking about global health for quite some time, but one thing that really sort of changed my course a little bit. So Lahore is a city, I don't know if anybody has been there. It's a city in the east of Pakistan. It's over 1,000-year-old city, really the best food you'll find anywhere on the planet. It's a city of tremendous culture, tremendous energy, and if this works, it's a city of sort of really happy people who are just living life to their fullest. And just a couple of years ago in this city, where my father was actually, my father is from that city, it's not very far from where I grew up, about 200 people died in a matter of a week, okay? And this was just, I mean, this was the biggest public health disaster in the country that I grew up in, the biggest public health disaster. In a matter of a week, 200 people dying. And it turned out that that was from substandard drugs. So we heard a little bit about counterfeit drugs, but that's not the complete picture. That's part of the picture. A lot of the drugs that are in the system, and I know there are many people who have decades of experience in East Africa and Southeast Asia, they can be just poorly manufactured. Nobody's making a buck out of that. It is just complacency. It's just incompetence. They may be degraded. And this would be sad and tragic if it was just one incident. Unfortunately, it's not. These are the stories all the way from Nigeria, where about 2,500 people died, kids, to Pakistan and US heparin incidents early on in this century. So what do we do about it? This was something very, very personal for me as well, because this was affecting my people. This was affecting something that was very, very profound, and we can come up with the greatest tools of biomedical engineering and deliver drugs. But if the drugs themselves are compromised, what do you do about it? So what I'm going to sort of refer to my students, then they'll talk more about this. And we'll see two separate stories here, one which is sort of slightly more advanced stage where we're going to the field, and one the early stage. Okay, so what is the process? How do we think about it? How do we sort of get involved with people from Ghana and Thailand and India and Pakistan in creating these solutions, devices, technologies as we move forward? I just want to leave with this slide. So if you look from the top, your top left, that's a picture that a friend of mine took in Lahore. Okay, that's a cough syrup. I mean, this should not be acceptable for anyone. Unfortunately, that's quite prevalent. On the right-hand side is a picture that I took last month myself. An expired drug that was going to a pharmacy in Ghana, sponsored by developed countries, UK, US, Japan. So they would send the drugs there, they would sort of just go and get rotten, and then eventually they would reach the market, right? So you are sending things with the best of intentions, but they're just not getting there. Something is broken there, right? So this facility had products from USAID, had products from DFID, had products from Japan, and there was no air conditioning despite the fact that there were vaccines there, right? It was 89 degrees there, 89, all right? At the bottom left is, if your eyes are better than mine, you can tell which one is real. I certainly can't. And you shouldn't be sort of forced to make that decision. And of course, the bottom right is you can get a drug of your favorite color, flavor, choice, shape, size, whatever. So this is what motivates people like us. These kinds of questions that involve technology, that involve regulation, that involve, as Patricia said, implementation, that involve public health aspects. So in many ways, what we're dealing with is a combination of these various facets of the public health sphere, and we are trying to come up with solutions that address these questions. And one of the things that was mentioned in the last panel, which I think is absolutely great, is that we don't want to come in with a solution. Instead, we want to come in as partners. We have a lot more to learn than we have to offer. And that is, I think, the essence of innovation here, that we come in with tremendous humility and sort of this eagerness to learn about the solutions that are going to work in that. So I'm going to leave there, and I'm going to sort of have my student, Darash, who actually has a prototype that I hope you'll get a chance to come and see after the thing, but I'll be happy to answer any questions. Oh, you want to go first? Yeah. Sorry. Well, this is not Darash.
Okay, hi everyone. So I'm Grace, not Darash. So I'm going to talk about, okay, so I just graduated from Boston University with my PhD. It was 5 years of work, and I was able to sum it up in 3 slides for you guys today. And I'm going to talk about my journey of working with basic science technology and trying to find applications for it in global health. Okay, so I worked with paper. Paper is a very cheap material, and in the last few years...
I think the slides are here, but they're not up on the screens.
Okay. So paper in the last few years has been the center of a lot of new research and using it for quantitative testing. So if we look back in history on the uses of paper, some of the cooler examples include invisible ink, using it to measure the pH of a solution, to separate solution proteins or analytes within a solution. And then in the 80s, using it for detection. So some of the examples include pregnancy tests where you can have a yes or no answer. And then where I started getting interested when I started grad school was with dried blood spots. So the example with the filter paper card and then the red spots, those cards are used to store infant blood so that way in low resource settings, so that way they can get shipped off to a testing facility where they actually have the resources to reconstitute the sample and then test for any HIV biomarkers. And so they found out that this is actually a very robust method of collecting the samples and storing the samples under conditions where you don't have refrigeration necessarily. And if you've ever worked with in a lab or just think about how to store things that have an expiration date, a lot of times you need refrigeration. And that's not necessarily available in low resource settings. So in 2008, there was a paper where they make paper microfluidic devices, which is shown on the far right. And they were able to create these tiny little tests that produce colorimetric results corresponding to different analytes in biological samples. So in this case, they were able to measure glucose, and then since then, it has advanced to become more specific and sensitive and to detect a wide variety of analytes in health and environmental monitoring and even forensic sciences. Next.
You can use the play button on the clicker, the green button.
So when I started, I had started looking at how robust proteins were stored on the filter paper, but I looked at different temperatures, conditions, and durations, but at the end of the day, I needed to find an actual application for this. And this is where Mohammed was really well connected, and he had somebody from, had worked previously in the Department of Health in Bhutan, and he was interested in the issue of road safety around the world, and in particular in his country of Bhutan, where there are several deaths due to drunk driving. And I was able to come up with a way to use paper in order to measure alcohol. And so if you just look at the current technologies for alcohol detection, there's a wide range of tests out there that vary in cost and sensitivity and portability. The one that comes to mind in terms of going to the field is the breathalyzer, which is in the middle. And even though that's really well used here and in several countries around the world, it's still not cheap enough in other parts of the world. So we wanted to come up with something that was even more low cost and just as sensitive. So on the bottom left are the paper tests that we built. This is on paper, so it's about the same width and weight of a sheet in the program books that you guys probably have. And the size of each test is about the size of a penny. And we got pretty good results with these. But the problem was that to get these results in measuring alcohol concentration, is you had to use this big dinosaur of a machine on the left, and that's called a potentiostat. It weighs probably like 20 or 30 pounds. It's really expensive. It's over $10,000, and this one turned out to be really old, but it still functioned properly. So obviously you can't take this out to the field, and that's one of the biggest problems with developing point-of-care tests in the lab right now is you can come up with something really small, really cool, and really low cost, but to actually use it, you have to use a company equipment that is totally the complete opposite. So what we did was we integrated this with a glucose meter. And a glucose meter is just a very simplified version of the instrument on the left. And when we did that, we were able to get good results and find a lot of interesting things in developing these paper-based diagnostics. So in a sense, we are basically tricking the glucose meter to measure ethanol concentration or alcohol concentration. And this served also as a platform for the measurement of other analytes. So we were able to use this to measure the concentrations of misoprostol, which is a drug mentioned before, to treat postpartum hemorrhaging in women. And I guess throughout this research project, networking was a really big part of trying to come up with applications for the paper-based method of storage. And it just meant going, like, talking to different people and making sure that my PI, Mohammed, told me his ideas or connected me with people, and also emailing others that I thought would give me ideas, and also coming up with my ideas myself. Okay, I'm gonna leave it up to Darash next to talk about his project.
Sorry, I didn't mean to cut you off there, Grace. My name is Dr. Darash Desai. I also just recently graduated from Mohammed Zaman's laboratory at BU. And going back to this problem of counterfeit and substandard medicines, that was really kind of the project that I was focused on there. And you know, talking again at the problem, just to give an example, malaria kills about 1 child every minute, yet 30% of malarial medication around the world is substandard or counterfeit. So, and that's only just one problem out of many. And so this is really a huge global issue. And there are a number of technologies that are out there that kind of are aimed at addressing this problem. But as Mohammed pointed out earlier, a lot of them are focused toward counterfeit medication. And what we wanted to do is not only look at those, but extend that out to these substandard medicines, the ones that are being manufactured, not necessarily with ill intent, but due to incompetence or due to kind of financial restrictions in production. And so what we developed at BU was kind of a new method of taking a look at what exactly is in your tablet and how much, specifically how much active ingredient is in your tablet. And that was really the focus of our project. A lot of the technologies out there right now that can be used for even substandard medications are mostly spectroscopy based, and they have a lot of really cool point and shoot kind of instrumentation. But it really gives kind of an overall fingerprint. And the big issue with this is that you really need kind of a huge library in the back to kind of compare results against. And the key thing that these technologies don't really address is exactly how much of the active ingredient is in the tablet. This is probably one of the single biggest issues with substandard medications. Impurities are also another issue, but really want to focus in on just how much active ingredient is in there and how can we quantify that. And so this was kind of, I don't know where the slides are. I don't have too many slides here, but what we really wanted to do in coming up with a new approach for this was to kind of take a step back away from the current technologies and rethink, you know, what is the best way to actually test this problem? And this really comes back to what Yusuf was saying earlier. That's a good point. Yeah. This is coming back to what Yusuf said earlier. It's really crucial for these sorts of projects that you do iterate from the actual technology development to user requirements specifically, and then to commercialization. And for us, user requirements was really a big key factor in how we were going to develop this technology. And at the time, I think we had a very unique opportunity where we were actually approached to kind of help address this problem by an organization called the United States Pharmacopoeia, who are actually based not too far from here. But they've, since 1992, been involved in drug quality testing around the world. They operate in over 42 countries. And they kind of help these different medicines regulatory authorities around the world address the issue of counterfeit and substandard medication through training, using instrumentations like the types that we developed here at BU. And so with the help of USP, we were really able to identify what the real key features were that we wanted to kind of pinpoint and tackle. And one of the biggest issues was just portability. A lot of these countries have sentinel laboratories where they have really big instrumentation, And one of the challenges was really, how do you come up with a quantitative instrument that can be used out in the field? So I won't go too much into the technology itself, but what we have here is just an early stage prototype where we were working with a fluorescence-based system, and we wanted to figure out how can we take that huge fluorescence-based system that usually requires lasers and a huge benchtop microscope, how can we take that into the field and do the same kind of analysis without losing any of that quantitative information? And so this picture here is just kind of an initial prototype. We went through multiple iterations, and as you can see here, this is actually 3D printed, which was great for us, and it was a great tool to kind of iterate through these ideas and really figure out, okay, what's working, what's not working, and how can we move forward from there. So through those multiple iterations, we were fortunate enough to actually, with the support of the Coulter Foundation, begin working with a product design firm, actually. And I think the key feature really here was that it wasn't kind of an outsourcing, and I think that was really crucial to the success that we had. This was very much a partnership. We went over there to the product design firm, really worked with them to figure out how we can integrate these components together and what kind of components we can use. And what we came up with here is kind of this portable system that we call PharmaCheck. And it's actually up here if you guys want to take a look later. But the idea was we wanted to create this integrated system. And so this is a completely self-enclosed system. It's got a LCD that's touchscreen that really helps the user kind of interact with the instrument and really eliminates the need for kind of really big manuals, because we have a step-by-step procedure that we can just run through one step at a time on this LCD screen. And so the user can kind of put in all the various components into this system and kind of just hit the start button. And that's really kind of the idea that we had, was how can we eliminate all sorts of sample preparation and make this device something that's really easy for the user to use. And I think that was, again, one of the really key features to the success of this project, was that instead of being technology-driven, it was really user-driven. So just to kind of reiterate, the key things that we focused on technically was that it was quantitative and had the potential to really not only look at the active ingredient, but how it's dissolved from the tablet. And through that, we were able to develop something that was less than 20 pounds, really easy to take around. The current field standard is 200 pounds. So it's a huge step in the right direction. And it's comparable in cost to a lot of the other technologies that are out there. And just lastly, we were able to take this out to Ghana just last month and kind of do a pilot test, and got some really great results. We're able to train users out there, and we're able to get a lot of the feedback that we need to kind of go through this iteration again and develop the next kind of model for the device that really hones again more on the key user requirements and how you can make this technology really easy for the user to use. So I'll leave it there. Thank you.
So let's thank the 5 mothers of our panelists for their hard work in bringing us this panel today. We have 10 minutes left before we are released, so I want to use this time for questions, specifically starting with the BU team. Could I first ask each of the innovators if you could answer in 1 or 2 sentences this sentence: my vision for commercialization and implementation is, Sean? And tell us which project, again, remind us.
So this is for the antenatal, the screening test. I would say my vision is to actually reach women not only in the health clinics, but also to their homes, where, you know, up to 50 to 60% of women don't even travel to health clinics.
Great. Darash?
So again, working on the counterfeit and substandard medicines. My vision is really kind of to take an integrated approach to solving this problem. And, you know, we're really focused on just the technology aspect here, but there are a lot of other aspects such as policy that really come into play. Education is another huge thing. So I think the vision not only is to get the product out there, but to also, in parallel, kind of work these other disciplines and come up with a concerted effort to kind of address this problem.
And Grace?
So my vision is to make the technology, so I work on the paper diagnostics, and my vision is to make the technology simple to fabricate and cheap enough to fabricate so that it's attractive for stakeholders in low resource settings to do the building themselves. So that way they're more closer to developing the product for the people in the area local to them that need it.
And my vision is that in 10 years, you guys will send me an email and say, I am so successful that I have all this extra money and I want to fund these other teams. Do you have any great teams that you can connect me to? All right, questions for the BU team? Yes.
I actually have a question for maybe all the panelists. Could you please comment on the regulatory hurdles faced for launching or actually even field testing these medical devices in low-income countries? Can you also contrast the situations with the developed world? Related to that, the question is, are there any specific challenges that are specific to the countries, and whether challenges can be overcome for devices that are not in the type 1 category, like for type 2, type 3, for low-income countries? Do you see that the hurdles can be realistically overcome in the realistic time frame?
So let's start with the professors. How do you teach about the regulatory pathway?
I think there are a couple of things here. One has to recognize that different countries have different laws, and there are certain countries where the laws are not very well defined. But the real sort of strategy that we have adopted is to take very early stage ideas to our partners, so that we build partnerships early on and we are not sort of surprised at the end when something new comes on. So rather than taking the final product and say, this needs to go through regulatory process, we have partners who are working all along with us, and we are very cognizant of the needs, the challenges, and the opportunities. So for example, in PharmaCheck, we are working very closely with the FDA Ghana, with the people who are acquiring medicines, who are regulating medical devices and medicines early on before we sort of really go into that stage. So that's critical in terms of that. The second thing that I think is important also is to have them give input as well as to what is needed and what wouldn't fly. Okay, so both in cases of us telling them what our idea is and them telling us of what does or doesn't work or where the need is. If you have these partners, I think the problems are actually resolved much more easily than if you come in at the end with the final product.
Yusuf, anything to add? That was perfectly stated. Okay. Great. Super easy, right?
Hi, my name is Kennedy Anene. I'm a physician from Nigeria, and I recently completed my master's in international health policy and management. My question is to the first set of panelists. First, I'll start with a comment. Having worked in developing country for quite some time, you see a lot of times people do bad, but with good intentions. For instance, in the screening of prenatal women, sometimes just because of lack of the facilities for dipstick, you get the dipstick strips being split in two just to increase the number of people you are able to use it for. Using your drops, do you have any, first I would like to ask, how does it compare with current technology that is on ground? Secondly, did you guys consider things like storage conditions, expiry dates, and things like that? And the issue of having listened to the next set of panelists speak, I begin to wonder about things like counterfeiting your product and how does your product mix with, because it may get to a point where one facility will run out and will want to share a bottle of the same reagent with another facility. So if you have a product that maybe is 1 year old and you have one that's just a couple of months old, how does it affect the quality of the test if you get them being mixed in the same bottle and things like that.
Yeah, so real quick, that was actually the inspiration for this project. One of my now advisors, who I wasn't affiliated with before, was traveling in these areas and actually saw the health workers taking those little dipsticks, which, you know, they're barely a centimeter wide as they are, and they were actually cutting them into thirds just so they would have enough. So we're certainly well aware of that issue. That was actually what inspired him to look more into this issue and eventually get us to this point. And we are, you know, we're definitely keeping that in mind. That's one of the reasons why we ended up switching from the pen to the dropper. Hopefully to help prevent that, you know, in the pen you can make smaller dots, which we wanted to try and avoid, so that does help a little bit having the dropper. You know, you can't have half a drop, you just put one drop on there. As far as cutting the strips, that is something we are still worried about. We are aware that it is an issue, so hopefully that will help us plan for it. Right now, what we're hoping is that with enough training, they won't do that. And also, hopefully with getting the cost low enough, they can, you know, keep a constant supply so they don't have to worry about running out. Are these perfect solutions? You know, we're not sure, but it is something that we are mindful of and, you know, going to watch very carefully.
So quickly, to answer your question about counterfeiting, the product that tests for counterfeiting, I think the issue there, or the real key, is being quantitative. If you're saying yes or no, it's a lot easier to counterfeit, but if you are able to provide a number, then you're able to say this is 90% pure versus this is 82% pure versus this is 51% pure. And that is one of the reasons we have sort of pushed hard, and having graduate students who are able to sort of really develop the technology to the point where you can get a number out. And that is very, very, very important. So that people can use it and say, look, this is not good enough, or this is okay. So that's one of the things. The other thing, there are a couple of parts here that I think are still difficult to be manufactured in low resource settings, particularly the microfluidic system. If you come here, we can show it to you. And I think that it's going to take some time. Finally, you have to use the same model and many others in industry use. You have to be two years ahead, because there will always be counterfeit. So you have to think of a 2-year strategy and come up with a new sort of a better improvement of that in 2 years. That's just how typically the industry works in many of these products. But before I forget, I totally forgot to mention, the next year's meeting is in Boston, so I hope you guys will be there. I'm promising you great weather in Boston in March. I'm promising right now.
And can we have a panel on implementation next year in Boston?
We absolutely will.
Okay, you heard it here.
Jennifer, I'll add it.
Yusuf, go ahead. Last comment.
I think there's one more question, Jennifer.
I'm sorry, we don't have time for one more question, but you'll come up after. Go ahead, Yusuf.
Just very briefly on PharmaCheck, what really excites me about the work that's going on at BU and the PharmaCheck is it ties to the issue of counterfeiting, it ties to the issue of regulation, it ties to the issue of corruption. I was in Guangzhou and I asked one of our hosts, what do you do usually when you go to the United States? They say, we go to Walmart and go shopping. I said, well, 99% of the stuff made in Walmart is made right here in this city of yours. And they said, but it's the quality control. When I buy a vacuum cleaner at Walmart, and if anything goes wrong with it, I return it. That means that the signal is passed up the supply chain that you better not sell this guy anything that has any hint of lack of performance, because he's empowered to return that right away. And they have notices inside the box, please, for the love of God, do not return this thing if there's anything wrong with it. Call us up, we'll help you work through it. That's what we should do for global health, because we're never going to solve the problems of corruption. We're never going to solve the problems of corruption within the supply chain or even in the factory level. But when you empower the end user, then suddenly 99% of that problem is solved. When the end user knows the potency of the drug they're giving to their patient or they're taking, I think that'll have a profound effect. So this is probably the most exciting and impactful project I've seen in a long time.
So I'll just close by saying that it's not the technology, it's not the invention, it's not even the innovation process or the educational process that supports all of this work. 90% of the blood, sweat, and tears is about the training, the implementation, the manufacturing, the marketing, the dissemination, the repair, and the constant iteration that you've just heard has to keep happening, not just on the physical prototype, but on the entire system. And that's why this work is so hard, because if it was just the technology, there would be no global health problems left, right? We wouldn't be here. We'd be at Mother's Day brunch. So thank you for your attention, your great questions. Call your mother or a mother if you haven't done so already today. And please come up and meet our panelists, look at their prototypes, and ask them questions if you'd like. Thank you so much.
Naturally